Well, general information, the QNet coordination team witnessed one change. You can see on the right top corner, Ilana Stein. Welcome to Ilana. She joined QNet, the coordination team, a few months ago and replaces Frederick Lenz, who resigned for personal reasons. So the QNet team is now even more diverse than it was before, I would say. And if I count the numbers, we are also quite balanced in terms of gender. So this is very nice. So welcome, Ilana, from all of us. What is new otherwise? Well, we already then had 2 QNet seminars organized. So this is a new series that was launched since the last workshop. And well, the second, just to maybe highlight something, for the second seminar, we already had 70 participants. Today, right now, I count 81. Hopefully we can increase this even, but it seems also this seminar series attracts quite some interest. Well, what I can say here, still to the seminars, we are always eager to learn from you, whether you would like to present yourself. Maybe it's an exciting study, a new topic where you think, hey, this is worth it to present in a, let's say, in a platform or in a way that allows for discussion. For those who participated, there is really plenty of time for discussions, and I think that's a very interactive platform that can be also attractive for many of you. Then, maybe last but not least, I also would like to remind you all about the QNet Forum. So this is really dedicated to allow interaction between our activities. If you have a question, if you have an announcement for an open position, please use the forum, and I'm pretty sure there will be someone out there among the right now 480 or 90 QNet members, so quite many. There will be someone who will reply to you, and maybe there is also an exchange there in a more written way. Before I hand over to the main organizers, you already saw that there was a message before asking you or making you aware that this will be recorded. So again, like in the 3 other workshops we organized before, we will record this workshop and make it available within the QNet member area. So this is all, and with this I would like to hand over to the main organizers, which is Alan Crivellaro, Ilana Stein, and also with some great help of Angela Brendin. Thank you.
Hello, everybody, and welcome to this workshop. Again, on my side, I'm Alan Crivellaro, and together with Ilana Stein, we co-organize this workshop within the QNet activities. Now, when we think about bark, actually, it's probably the most common scene we have when we walk through a forest, right? We see bark on tree stems and everywhere, up to the crowns, down to the roots. We should not forget about that. We can make the bark story very simple. It's just everything outside the cambium, but we actually know it's much more complicated than that. I don't want you to go through the terms and the figures in this slide. I just want to highlight how complex the bark is in general, because of its very extreme variability within plant species and between different plant species. If you climb a tree along the stem and you study the bark there, you will see a lot of anatomical changes related to the distance from the top, to the plant height, and also to the age of the bark. We don't go into that now. We will do it via the speakers through the workshop today. To introduce a little bit the speakers, I want to give a very brief overview on bark. We should start, of course, from the vascular cambium, which is the tissue originating the wood on the one side, and today we are on the other side of the cambium towards the bark. Well, there is another cambium, often called the cork cambium, which is located within the bark. And this cambium is actually called phellogen. It produces also 2 different tissues, phellem and phelloderm, towards the outside and towards the inside, respectively. And these 3 tissues all together, they are called the periderm. The periderm then is part of the bark. It's definitely terribly important for allowing some of the functions of the bark. And we will have Simca Leviadun explaining us the secrets of peridome structure in woody plants as a first speaker. Of course, the cambium, the vascular cambium now, the one originating the wood, is also originating phloem on the other side. And this is also a terribly important tissue for plant functioning. You can recognize it very easily because it contains rays. If there are rays in the wood, there are also rays in the phloem. And on this side, we will have Joseph Glitzer. She will tell us something about phloem phenology and structure in tree from different environments. Something related to changes, which is a kind of a key word when we study bark. When we study bark, or when we study wood, I would say when we study wood in general, the bark just fills the gap in between. When we collect samples, there is the bark there. We are not interested in it, so most of the time we remove it if we only want to focus on the wood. If we don't remove, we know it creates problem because maybe it sits on top of our sections. We don't want that. Even if we want to study the bark, then we know we have to change a little bit our way to cut thin sections compared to the wood side. The tissues are of different hardness, and they are in the bark very hard and very soft tissues combined together. This is why methodological issues usually arise when we want to study bark. And so we invited Marcelo Pace to explain us methods in bark anatomy research, and to again to tell us the secrets on how to get good sections, which are of course the basics of a very good quantitative bark anatomy study. Then we will have a break, but after the break, we go into the application. Then bark variability is something I already mentioned 3 times now. And as I said, it's kind of a keyword of today's workshop. And what's the function of the bark, knowing that it has so many different structures in it, and these structures, they change quite a lot. And this is something which will be discussed by Julieta Rossen. On bark functional ecology is the title of our talk. And we are really looking forward to learn applications on this side of bark studies. Another question which is very often asked from students, but also from bark experts, is what's the relationship between the anatomical structure of the bark and what we see outside on a tree stem? Well, there is a lot there, starting from terminology, but also getting into the deep relationships between bark anatomical structures and its macroscopic appearance. So Alexei Oskolsky will explain us again secrets about these relationships. So this is the panel of experts we have today. They will all speak to us for half an hour, according to this program. You have 2 ways to interact with the speakers. One is writing in the chat any thought or question you might have during the presentation. Then Angela Prendin will mainly collect all your questions and what you want to learn more about the topic which is under discussion. And at the end of the workshop, in about 2 hours time, we will put all of them together and we discuss with all speakers, and we'll ask them to answer your questions. During this time, during this discussion time, at the end of the workshop, you will also be welcome to share your voice and ask questions live anytime. That's all on my side. I'm now stop sharing my screen, and I invite the first speaker, Simca Leviadun, on Perida.
Do you see the?
Yes, we see it.
The first thing. Okay.
Yeah. You should enter presentation mode.
Excuse me?
You should enter the presentation mode so we can see the image on full screen.
I did share. Where is the presentation mode?
at the bottom right down there, there is a screen-like thing to the left a little bit. I see your arrows, so I'm trying to guide you.
Try again.
A little bit lower down. And
Lower
then left.
down.
Yeah, and then left. Yeah, down there, straight down. Oh, I don't see your pointer anymore.
No. Is it up here? Now it's
No.
okay?
No. You see
Yeah.
down at the bottom right corner, you have this plus minus the zoom.
No.
To the left of it.
No, I don't have it.
Yeah. Or you press F5. Try to press F5 on your keyboard.
F5. Yes.
Oh,
Pressed it.
didn't work.
You see it now?
Oh, we see it, but not full screen.
Full screen. I'll do it full screen. You see it now better?
Oh,
yes.
No, yes.
Okay, thank you. When I try to explain things to current, mostly young people who deal with plants, I always have in mind my postdoc, which was molecular. As an organismic botanist, I decided to have 30 years ago a molecular postdoc because I thought that I cannot be a good botanist or any type of biologist without having a real experience in molecular way of thinking, knowledge, and methods. And I always have it in mind when I go in the field and cut my samples, 3 samples with a handsaw. Because I found huge ignorance by very talented people. It's not their blame. And they were taught in many times by people that don't know botany. They are called plant scientists. Those are people who don't know botany and deal with plants. And for instance, I know universities where people teach that the pit in the primary plant body is the outcome of cambial activity, which is of course not true. So for those who are very experienced in plant anatomy, please forgive me if some things you know already. It should be so. The cross-section of a young tree you see here is Quercus suber, the cork oak, and one can see the cambial region, the cambial zone, the phloem, which is quite dark brown, and lots of cork. This is primary cork, and in this important species for cork production, for the phylogen is active throughout the life of a tree, which can be hundreds of years. And in commercial plantations, it is peeled. The cork is peeled after 20, 30 years. And the phylogen continues its activity. And we get, again, a nice production of cork that will be harvested 20, 30 years later. But not all plants produce a single phylogen throughout their life. There are many species. For instance, this is a broken part of the cork of Pinus halepensis, a Mediterranean pine, that shows layers of cork produced again and again, and it's called ritidome, which means after the phylogen is active and produces whatever it produces, and it's more than one type of cells. In this case, it's not only phloem cells, but also stone cells, sclerites, that make the bark more secure towards mechanical and biotic dangers. And after a while, cells that were differentiated within the bark change their fate, go back to the cell cycle and make a new phylogen inner. And this phylogen with the core produces cork. The cork is impermeable to many factors like fluids and gases, and the tissues outside it die. And so the cork in this case is made not only of phylogen derivatives, but also of some cambial derivatives that take part in the layers of periderm. The phylogen can initiate in various types within the bark, various cell types and layers within the bark at various distances from the epidermis. In this case, we can see the initiation, several cell layers here. Here is the epidermis with a cuticle. It's 2 or 3 cell layers deeper, but there is a variability. And if we check different conifers, as I did many years ago, we can see, for instance, at number 5, that it started only 1 cell layer beneath the epidermis. Here, 2 cell layers at number 7, and number 6 even deeper. And if we go to a cross-section in a young stem of Cupressus sempervirens, we can see the cork, the new thin layer of cork. Sometimes, like here, it's formed at the upper right, formed just beneath the epidermis, but some in other places in the same cross sections, it can start quite deep, cutting out various tissues. So it's flexible. If we leave the conifers and we go to Dicotyledons, Calotropis procera, which is a desert tree or shrubs, depends on growth conditions, found in India, in parts of Africa, in the Middle East, in arid areas, and I think also even in Central America. We can see that it doesn't start all around the stem. Here we see the beginning. Here it started earlier and we can see more cork. And here we can see more cork, very soft and brittle in the case of this species. And it isolates the photosynthetic tissues from sunny radiation. If we look at a little bit older stem of this species, first we can see lots of gum ducts. This tree is well defended also by quite toxic white gum. Although there are some animals, invertebrates that can consume it, I don't propose any student to do it. And we can see lenticels from time to time. And here, when it became older, it covers all the surface. There is a long-held belief based on papers which are now about 60 years old, that the pressure in the stem, because of quick growth, causes the differentiation of cells into phylogen and to form more cork. This is a mistake. For instance, if we look at this olive tree, At the left, we see an old branch at the diameter very close, very almost identical to a younger branch that emerged from the older one. Many years after the old branch was formed. It's very common in olive trees. And in the old branch, the cork is thick, although it grew slowly. And in the new one, which grew very quickly, there is almost no cork, only a very thin layer close to the surface. And if we go to Melia Zadarech, where I did also sections, at the left, we see a thick stem that grew just above a water and a source and grew very quickly with broad, very wide root rings. And we can see an older tree, much older, at the right, which much thicker cork, or peridem, if you want to call it, that grew in the same village, but differences in growth conditions. Now, in a cross section of the one who grew fast and had at least pressure, mechanical pressure in the tissue, there was only the primary periderm, only one layer of phelogen that produced some cork, and this is it. And the one who grew slowly, we have here the arrow points the innermost lepidome, and then there was one who was formed previously, and of course there was one outside. So this hypothesis should be taken with a grain of salt, since nobody checked enough plant taxa under enough growth conditions to know if it's a rule. But this thing that was published about 60 years ago concerning trees in Indonesia might not be the true picture, or at least certainly not for all plants. When we try to see what factors control peridem formation, and if we look at trees or woody plants or perennial plants, not only in cross sections, and I think I have the pile of papers I have about cork starting from somewhere in the middle 19th century till today. I think now it's almost a meter tall, the pile of reprints, and the vast majority of them is of studies who used cross sections. But if we look at the plants, at the whole plants, in the field, in the forest, even in the greenhouse, and you look for patterns, we can see, I saw it in many dozens of species belonging to various families, various types of tropical and non-tropical and temperate and deciduous and evergreens and whatever. We can see around buds and sometimes beneath them. And beneath the leaves, or sometimes specifically beneath the major veins that go out of the leaf, we see that cork is not formed, or much less cork is formed. And I can say in advance that it seems to have a function, but we can learn something about the regulation. And this was, the previous one was in Ficus religiosa, which is an Indian plant, planted as an ornamental all over Israel. And this is Ficus elastica from a different origin. And we can see the same phenomenon. very nicely. And if we go to another plant, which is planted in many hot countries, Melia azadarech, we can see that when this deciduous tree has no leaves in winter, we can see less lenticels and less cork under the major vein, and again, less under the quite large veins at the lateral parts of the leaf that existed there some time earlier. And if we go back now to the Calotropis procera, which, as I said, is a desert plant, we can see here is a bud, here was a leaf, no cork. Again here and here above, you can see no cork here and no cork there, because the leaves are alternate. There are two, and they alternate, and always underneath the leaf and the bud, there is no cork. This is the rule, but the rule can be changed. And this is a close-up, but leave it. This is a sketch I did from a branch which was more than a meter long, and where there was cork. I did it on top of a picture. It's black, and where it was green, here it's white. But this can be changed, because this branch grew there when it was directly exposed to the sun. It's the Dead Sea region area. Summer temperatures are 40 degrees Celsius in the shade in a cool day, and it can be 4, 5, 6, 7, 8, 10 degrees higher in the shade, and those branches are exposed to the sun. And it's known already from the 19th century. For instance, I think studies by Dulieu, that also in European trees, the side that is exposed to the sun, directly to the sun, can produce cork earlier than the shaded ones. And now a little bit about the regulation. We go back to Melia azadarech, young trees. Here was a leaf that I broke it. We can see lenticels here and here, and no lenticels here, and very few here in the side, related to the major veins. And here we can see more cork, and also there were more cell divisions and huge dilatation in the bark, because I applied ethylene to this place. And I want to say something about regulation. It seems that auxin, that always goes from the buds towards the roots in a polar way. It delays cork formation, and where you have less auxin, you have more cork formation. And when you apply ethylene, you have more cork formation. And if you stop the aeration of a stem, as it was done in experiments without thinking about the hormones more than 50 years ago in Eucalyptus camaldulensis, which is very sensitive, we get cork phelogen initiation and cork formation very quickly. So altogether, and the same is when we have lots of sun irradiation or wounding, because that wound ethylene and stress ethylene are produced easily, and some plants are very sensitive to this. And from unpublished data, I can tell you that not all plants are sensitive similarly to this. And I can say quite safely that auxin inhibits cork formation to a certain level, and ethylene induces cork formation because ethylene induces many types of defensive mechanisms, and cork is one of them, a very important one, because if enemies or physical agents cross into the plant, the damage is big. And the periderm, after the plant becomes woody and the cuticle is less important or disintegrates, periderm is one of the first defenses of plants against various troubles. When we look at fast-growing organs that have more auxin than slow-growing organs usually, we can see again that there is a delay in periderm formation, and in leaders or branches that grow quickly, the cork is formed, the phelogen, and the cork is formed at a larger distance from the growth apex than in slow growing ones, or towards dormancy in winter in cold countries. And now I want to say something about the probable function of delay of cork formation around buds in so many species, as I said 10 minutes ago. This is Melia azadarach, which is a wonderful plant for experiments because it's very hard to kill it. Whatever you do, you get new ones. You can have new trees from major roots, and if you cut the trunk or cut huge branches, you get regrowth. If the cork is thin and there are still many openings where there were buds just above the leaves, which is a rule in many plants, you get branches along that young trunk or branch that was cut because there were buds ready. They didn't have to redifferentiate via callus as it's in older trees with very thick bark, and grow quickly and help the plant establish its position in the canopy in the competition for light in the forest. If the tree is older than what we see at the left, as it is a medium age at the right side, here and there in a crack that had very thin cork, the branch can grow quickly, and again, it helps regeneration. But if the cork becomes very thick, with many sclerites, let me go back. Oh, here it's under polarized light, it's Melia azadarach. You see the sclerites and the fibers, and it's fibers. And it's very hard for a young bud to grow through bud. And then the only way for a generation is to form callus here at the cambial zone or very close to it, and then differentiate into new buds, and the buds grow a wall of even age branches, and they start to compete on the weaker ones. something that takes several years, or sometimes even many years. And it's not easy to regenerate and get the place in canopy because this process is much longer than picking of suppressed and turn them into photos.
Simcha, sorry, you have
Yes,
5 minutes
I know,
left.
5 minutes. I know.
Okay.
I have my clock in front of me. I am almost done. We can have funny things in cork, for instance, in this case in Melia Azadarech, but in many other plants. Cork fingers. This is the bark, and in the bark there are bands of fibers formed by the cambium, secondary phloem fibers, which are very common, and from various plants we make fibers for the industry, or for the paper industry, or for fiber industry from those fibers. And as I did it in experiments in Melia Azadarech, I wounded the bark with a sharp razor. And here you can see in red the fiber bands that we saw previously in a cross section. The fibers were exposed to the air. The air went into the bark and dried it up and caused stress. And then there is a differentiation from various parenchyma cells of the bark, a differentiation into phellogen, and we get wound phellogen, which forms fingers that go up till the tissue is not so dry. And then it forms, if you look at in a cross section, you see rings of traumatic cork formed around the fibers that introduced dry air and had the potential of introducing fungi and other pathogens into the tree and blocking this dangerous area from the potential of first desiccation and second pathogen attacks. Okay, I think it's enough because I don't have time to go growth, which is another type of very important meristematic activity. I think it appeared very early in evolution. And it goes hand in hand with the phellogen, because I'll make it short, of course, because there is no time. And again, ethylene, either by wounding or by external application, is inducing lots of dilatation activity and the formation of secondary, tertiary tissues, because dilatation meristem, or just dilatation without a meristem, and both processes occur sometimes even in the same section. Therefore, the dilatation starts from secondary tissues, and that's why maybe we should address it as tertiary type of growth and not as secondary. Thank you very much.
Thank you very much, Simcha, for a nice lecture. I want to ask everybody to ask questions in the chat, and we will discuss in the end of the presentation of this workshop all the questions that were asked. Now our next lecturer is Jožica Gričar, and she will speak on phloem phenology and structure in trees from different environments. So welcome, Jožica. And
hello, can you hear me?
Yes.
Okay, good.
Super.
In the presentation, you can see it? Yeah? Okay, perfect, thank you. At first, I would like to thank the organizers for inviting me to have presentation on the phloem, the studies that we have been doing, I think, for the almost last 20 years. And you can see from the title, I'm going to talk about phloem phenology and structure in different tree species from different environments, mostly from temperate and Mediterranean, submediterranean. As it was already mentioned by Alan, I will talk a lot about seasonal changes, the effect of phloem phenology on the structure of trees. So you can see here I have many points. Well, if I will not have time, I will just mention what in addition to seasonal dynamic of phloem formation and structure should be considered if you decided to study the phloem part. Here is one illustration, as it was also mentioned. Bark, actually, we can talk about all tissues outside the cambium. Vascular cambium, and maybe just a point of this IAVA list of microscopic bark features, which I think it's very helpful, not only for beginners, but also for us sometimes to check if the terminology is unified. Bark structure, I'm going to focus on this very narrow part of the bark. This is one example of Quercus petrea. I will talk about uncollapsed phloem. More precisely, I will talk about mostly about the youngest phloem, which is also a narrower portion. I would say it's less than 10%, about 4 or 5% of the entire bark tissue. When we started to study xylem formation, at one point we were actually thinking that we are following radial growth. So by that, I mean the activity of the vascular cambium. But here you can see that illustration that radial growth, it's not only the activity of the vascular cambium, it is much more than that. So we have another cambium in the bark, as already mentioned. And then we cannot exclude secondary processes that are going on in the bark, are age-related, season-related, as for instance, scleroid formation, collapse of sieve elements, inflation of parenchymas, and so on. And of course, all these processes, they have effect on the radial stem changes. So mostly the analysis on radial growth are focused on xylem formation. Of course, I think that the main reason is in the endocrinology because it has the highest endocrinematological potential. So we have these archives of 3 rings. But lately, we are also aware that the vascular cambium activity, it's not only the xylem formation, but it's also the phloem formation. So if you want to study the effect of different environmental factors on the radial growth of trees or the cambial activity, you cannot ignore the phloem part just because you think that maybe it's not important. And I think in the seasonal studies, we need to look in the cambium as a whole, so on the xylem and on the phloem part. So sometimes also in the literature, it was statements that maybe the phloem is not, it doesn't have so much potential in terms of dendroecology or dendroclimatology. So it means that it's more stable in structure, it's more endogenously controlled, and it means that it's less dependent on environmental factors. And well, I will show you in continuation that this maybe, of course, it is true in some parts or to some degree, but there is also a lot of information in the different phloem traits. What is also maybe a bit different, as in the case of xylem, is that in the phloem, the increments they conduct, so the annual increments they conduct only or perform its function only over a very short period, so maybe 1 or 2 growing seasons. It is totally different than in xylem. And after that, the tissue, it is exposed to different changes. So it's collapse, the function, the conducting function is not there anymore. And that also means that for trees, formation of the phloem every year is very important. So you can find some kind of locally missing rings in the wood, but you don't find them in the phloem, because trees, they need to rebuild the, let's say, highway in the phloem to link leaves and developing tissues. And here I want to show you some of our some results of our studies we have been performing. And here I show we had 2 temperate sites of in Slovenia, 2, let's say, economically and ecologically important tree species, Fagus sylvatica and Picea abies. It is kind of temperate forest, very optimal sites for tree growth. And in the last years, we have 1 site in Submediterranean, and we have different species there. Most important is Quercus pubescens. And for the nearly last 10 years, we have been also monitoring radial growth in this area. It's very interesting in the sense also how to study extreme weather events on the growth of trees. This is also this area, or this kind of conditions are foreseen to be to spread in Slovenia, so to go also to other regions. And we have in this area we have very common drought events, but also fires. So it's let's say for studying extreme events, it's very convenient area. So I'm not going to into detail about sample preparation, how we do. You can see also our latest video, how we prepare the sections, or you can also contact me or check our papers, how we do it, because I don't have time here. Now I would like to show you the main findings we observed in the trees from temperate environments. So basically it means that these trees, they have typical seasonal pattern. We have dormant season, and then we have this transition to active, so to growing season, and then vice versa again. And of course, because there are different environmental conditions, this affects morphology of the cells, of the phloem cells, and you can really find the annual growth ring boundaries in the phloem. So I see it here. It's one example in Abies alba, and this is another example of Quercus petrea. You see clear structure. We have early phloem. Here you have axial parenchyma cells. It's transition from early phloem to late phloem. And in the case of oak, for example, you have this clear presence of fibers, of the phloem fibers, which is also under transition from early to late phloem. So although the structure is a bit different, I would say that general pattern is quite the same. Usually the initial early phloem sieve cells, they start to grow before cambial's productivity starts. And then you have, in the case of sieve cell formation, you have different steps. So first the cells try to grow. And in the case of spruce or silver fir, you have also secondary wall formation. It's not the same as in wood, but it has a bit wall thickening. So you can see this, effect or situation also in the case of sieve cells. So we try to develop different, let's say, milestones of phloem formation through the growing season, and try to observe also structural differences. And so in the case of trees from temperate and cold environments, what we have is quite, let's say, similar developmental pattern. You have, as in the case of xylem, you have onset of growth, you have then a peak, and it is more or less at the same time cessation of cambial activity. So as a result, it's a typical, there are typical xylem and phloem increments, which can be, let's say, quite easily seen on both sides of the cambium. So at one point, we started to include observations of phloem formation in our data set. Let's say at the beginning, without any expectation to what are we going to see. And then when we had the data from 3 years, from 2 different sites. One was also in the Czech Republic. What we have seen is that actually that we didn't find so much differences in one location over these 3 years, but we see differences in the structure and width of the phloem among these 3 different sites. And the differences were mainly observed in the case of late phloem. We also measured the sieve cell sizes, and we also see some differences. So our main conclusion here it was that although they are quite optimal sites for tree growth, obviously the phloem structure and width is also, let's say, kind of plastic for trees try to adapt the structure to function optimally in the given location. But as I said, we saw a certain plasticity also in the phloem structure as well. Here are two examples of spruce and beech. So this was then our question. We saw these differences in the structure of early and late phloem, and then we also see seasonal pattern. So our question was how this can affect the potentially conductive capacity of the phloem. We know that it's not only the latest phloem growth ring which contribute to the conductive capacity, so we thought that we need to observe the entire non-collapse part of the phloem. So it's not only the last ring, similarly as in the xylem, so where we have a septwood, right? And so also in the phloem, we say we need to check the structure of the entire non-collapse phloem. And our idea was here because we saw many papers on the phloem in the last years. And my question was how we can compare our data, because I already knew that it's not so simple. It really matters if you take the data from in middle of July or in dormant period or even in spring. So you cannot just put, you know, the data together without knowing what you took. And so we did our example with my colleague Peter Prislan we just tried to, let's say, reanalyze our data from our archive. And we selected 3 temperate species of different porosity, spruce, beech, and oak. And what we saw, you can already see in the images, it's from the onset of cambial growth. So here, then you have the height at the end of cambial cell production and at the end of, let's say, a wood formation period. So a month later than the end of cambial cell production. And by naked eye, you already see that there are differences. So you see the differences in the share of the current phloem increment and the increment of the previous year. And when you put it into the scale, we saw that although we saw the differences, but there is kind of common pattern for all these 3 species. So that at the beginning of the growing season, what is expected is that the increment of the previous year is very important. So the trees really rely on the increment formed in the previous growing season. You see here, it means previous early phloem, previous late phloem, and current early and late phloem. And so in the end, the situation is totally different. And the share of these, let's say, 4 different tissue parts in the phloem changes. And also, if you look at the absolute values, you see that the absolute values are different. So that the widest width of the non-collapse phloem is in oak, and the narrowest is in beech. It makes sense because beech has no dead bark layer, so it has kind of space saving strategy, try to, you know, to optimize the structure to function, because it needs to crush or to store all the tissue within the one periderm. And so what we also saw that the width of the non-collapse phloem vary during the growing season, and it is the effect of 2 processes. So we have positive effect of cambial productivity, which contribute to the width. But then we have this negative effect of the collapse, which actually it affects negatively to the width of the non-colapse phloem. So here, I think that the main message would be that really the time of the sampling, it's important. I mean, it is logical if you do the research that you take care of it, but I see the problem when we try to compare our results. So we also measure the SIF elements. I'm not going into detail because I don't have time, but of course this early phloem it's more the SIF elements are wider. And I forgot to mention before that what we saw is that structure of early phloem, it's quite stable. So the main change is what you see, it's in the late phloem, while the early phloem, it's quite stable. So 3 to 5 early wood SIF elements. And then if you look in the sub-Mediterranean side, you know that here you don't have true dormancy. So we have quite different patterns in the case of xylem, and in the phloem, we don't know exactly what is going on. That's why we don't have the blue and black line. It's just our assumption. And as a result, you don't see annual increments in the Mediterranean climate in conifers. You don't have this clear band of axial parenchyma cells. You have, something. So in Mediterranean conifers, what you can do is you can focus on the analysis on the entire non-collapsed phloem, because also you don't know when the cells are actually formed. And it is seen, or it is suggested that the cambial activity on the phloem site, it doesn't stop, or maybe it stopped for some period, but they're not clear patterns. So the cambial rhythm in Mediterranean climate is totally different. And it's one example we did with the colleagues from Italy and Spain. So we took their two Pinus pinea, Pinus halepensis. What we saw also, again, the differences in the width of the non-collapsed phloem, also in the sizes of SIF cells, meaning that somehow they are affected by environmental factors. And it's interesting that usually the narrowest were during drought period. Also, the non-collapsed phloem is the narrowest here, but the frequency or the frequent number of parenchyma cells is the highest in the drought period. So the structure is somehow really, adapted to the environmental condition. Another aspect which really greatly affect the structure and the width is the vitality tree condition. So in the trees that are not so healthy, usually the phloem increment is composed entirely on the early phloem. And then, of course, I guess, I never checked also maybe the width of the non-collapsed phloem changes. So the structure, which actually it contains potential conducting capacity. And so sometimes I see the papers that say that the trees that are less wide, they have wider increments. It's maybe just because they don't have late phloem. So the structure is different, and there is a question what they can do if there is a lack or not enough share of axial parenchyma, if maybe this also contribute to the mortality. It's the same in spruce, or not in spruce, in fir and oak. And then one example that if we study radial growth of trees, soil properties also have effect. We have one plot, which we are lucky because it has the same environmental conditions, but totally different soil. We have limestone, very poor soil, and then we have very rich soil. It's one edge, very nice, so Quercus pubescens. And what we also saw is that the increments, xylem and phloem, on the let's say rich soils, they are much wider. So they can also affect, so exactly the same climate conditions, but soil really contribute also to the final width. And similarly, also the structure and width of the increments in different tree parts, it can also vary. It's one example again in Quercus pubescens from stem and branches. In stem, the cambial activity was 3 weeks longer and resulted also in much wider, in stem, much wider increments in the case of xylem phloem. So also, the position in a tree, it affects. How it's in the roots, I don't know. And then another example which I would like to show is that we have effects of extreme weather events. We were lucky that at once we also had a fire on our plot. It was at the end of the growing season, but our question was how it's going to affect the structure in the next growing season. And again, it affected, I'm not going into detail because I don't have time, but it affected, and actually in this case, the phloem and xylem increments were wider. So because I think that the vascular system was a bit somehow of ruptured, in the xylem we saw tyloses. In the phloem, I guess it was quite damaged. So wisdom, and also what we saw is that the share of axial parenchyma was much wider. And so lately, what we are also doing, we try to combine our phloem data, of course, with the xylem data, but also with leaf phenological data, because, of course, these systems are connected. And as I said, leaf and cambial phenology or xylem phenology, it's really connected to the phloem. So I think it's logical to involve these observations in our study. And it's just one example from 1 year for 3 different species, what we also observed the pattern. But what I like is that it's only 1 year, then you have sometimes more questions and answers. But now we have for Quercus pubescens, we already have 10 years of data. So it's multi-year phloem data, which I think it's very valuable, because then you can really try to look in this time year-to-year variation in the structure. You can compare it with other phenological data like leaf phenology and cambial phenology. And what we see from this data is, although we see from year-to-year variation, but the timing it's fixed. So so it means that in the case of oak, onset of cambial activity is always before bud break. And then the time of full leaf unfolding is approximated at the same time as transition from early to late, from late wood or early from late phloem. So this is, I think, it's very valuable. You can also see the legacy effect of the previous growing season, how it affects, you know, and this is something that it really can opens a lot of answers on how tree responds, first of all, to different environmental triggers, to extreme weather events, and to understand also a bit more, maybe mortality patterns in some areas of, you know, when the trees are dying. I think that the phloem can help a lot in this sense. So if I conclude, I don't know, I'm too long or not, I'm not. So maybe the conclusion that phloem is overlooked in the dendroecology, it's not true anymore, because there are many, many good papers in the last years. I think well, we need to say that the studies on the phloem are 100 years old, so it's nothing new. But I think that benefit now is that we really have good tools. We have computers, we have digital data, we have image analysis systems. And we are, in this sense, we are very rich, and it's the time that we can combine the data to get, you know, to more global, to go into more global areas, to better understand how the trees really respond to environmental or any other factors. And I think that 1 year in this sense, it's not enough, but multi-year data, different sites are very important in this sense, because as I already demonstrated, if you want to make any conclusions in trees from temperate sites, which are very easy, it can be very misleading. And then what I think is very important, or what is really missing, is knowledge about tropical environments, tropical trees. We don't know anything. I think that there the story is even much more complicated. And here, when there are many studies on phloem anatomy, I think that it's time to unify methodology, not to give some wrong conclusions. And I see also huge potential with development of quantitative wood anatomy, also to move on the other part, so to the phloem. And I think that these tools, they have really huge potential also to try to work maybe in the collapse part of the phloem, which is, it's maybe not important for conduction, but for storage for certain ease, because it's a lot of storage there. And I think there are very important and open questions to expand here and try to combine the data to get maybe a better picture on the tree response to different triggers, let's say. Okay, thank you.
You have 5 more minutes.
Well, I finished, so maybe we can, if there are questions, we can start now. I don't know.
Yes, thank you, Joyce, for your presentation. It was really dense and kind of quick. I'm sure many of you, they have questions, so I would like to invite you to write those questions on the chat so you don't forget them, and I think we will discuss them at the end of the workshop altogether. Joyce, you didn't mention methodology because you said I don't have time to go through that. I know you have a very nice video showing the methods you use, a very recent one, but we are also lucky. We have Marcelo Pace next, speaking about methods in preparing bark for anatomical studies. So I invite Marcelo to share your screen. Are you still with us, Marcelo?
Yes, I am. I'm just looking for my window here.
Good.
Should we wait some 5 minutes so people maybe can have a break or something before we start? I don't know if people need to.
Well, we will have a break after your presentation, so I will still keep the program as it is.
Okay, good.
Thanks.
Should I start?
Yes,
then?
Feel free.
Okay. Okay, so first of all, thank you very much for QNet. I think this is an amazing initiative. We have over 100 people here, and it's just like incredible. And so I'll speak about my experience in methods in bark anatomical research. And of course, like we are over 100 people here, and I'm sure that everyone has their tricks and tips. So what I'm showing is like what I do and what has been working for me. But then, of course, adaptations are always welcome. And of course, it's the first time in my life that I speak about methods. So if I am going too fast in some part of something is not clear, you can ask, or you can also reach out directly to me, either in my website or by email. But mainly in the presentation, what I will try to do, let me see if I find, and I'll try to go through the same processes that I would do in nature, like when I go and collect a plant until up to the moment that I have the slide. So start with the collection of barks, what has been working for me, and the fixation method, softening, embedding, and sectioning and staining. Of course, not always you need all these steps for every bark. So many anatomists go directly from the field to a section and it works just as fine. I won't speak about the paraffin method, but many colleagues use paraffin as a method and it works perfectly. And I think something that I did not include here in my presentation, but it's the most important thing, is the most important things is like always patience when you're sectioning barks and a very sharp knife. So these are the two like most important things probably. So of course, like when we have a piece of stem or root, our goal is to have something like what we're seeing here, right? So also we want to have the overall bark, so everything from outside the vascular camion, as we said before. So here we have here the phloem, and we have the sequent periderms forming the ritidome. And just comparing to what we saw first, just before the presentation, before, you can see here is a phloem without any collapse. When you have a lot of fibers, the sieve elements do not collapse. But the thing here, like, so focusing in the technical part, this is the ideal thing that we want. So the overall image, and also be able to see all the details, like we see here, the cambium, the sieve elements, the phloem, if crystals, everything is perfect. And this is from Ray Everett's collection. So Ray Everett was one of the most important person in phloem research. He was a student of Catherine Izzo, and he used methods that unfortunately and in part are no longer available. So he would soften with HF, which is highly toxic. I will speak about that a little bit later. And he was embedding in celloidine. Celloidine is nitroglycerin, and it's prohibited by most armies around the world because it's explosive. So we no longer have that as available. I know that Tomlinson still uses palladium. That is a good substitute, but I have been trying to buy for a long time and I can't find it. So, but also it was not a very practical method. Celloidine is very slow and very dangerous, of course. If you had all celloidine in your lab and you like the window closed too strongly, you could explode the entire lab. But then moving to the step-by-step process, the first thing is, of course, the collection. So unlike wood, many of us work that you can keep in a wood collection, and after 70 years, you go there and you take a piece of wood, rehydrate and section. That's not the same for bark. So usually you need a fresh collected material to be liquid preserved. So here you can see a photo of us in the field. I work mainly with liana, so the damage is much stronger, but the idea is always to try to take a section from the stem or root without killing the organism. You can take a branch, of course, but yeah, so the most important point here is to preserve in liquid. And usually what we do is we have more than one option of preservation liquid. FAA has a very bad reputation nowadays, and people say that it's not as good as many others, but the section that I just showed from Everett collection was fixated in FAA. So I think that the step that come after FAA may be more important even than the fixative itself. But if you have a large sample and you cannot make a smaller sample, or if you want to see the overall structure, FAA is really good. But if you really want to go into details of the cell, organelles and everything, then maybe you should choose another type of fixative like CRAF, glutaraldehyde, and the conservation is typically in ethyl alcohol and ethanol. All the things that I'll be speaking about, I have summarized also in this chapter that I can send to whoever wants in this book. But here, I just want to show an image of how different it is when you have a liquid preserved sample and when you let it dry. So here you can see that although the xylem may be well preserved, the bark changes completely. So when you try to rehydrate this and make the sections, you will already have lost a lot of the information that is really important in our studies. One option, as I said, starting the presentation, is to section fresh. That's always the best option, actually, if possible, either fresh or directly from the liquid preserved material, the pickled material. And you see that that's the best method because the cells are in perfect shape. So you can see the sieve elements, the parenchyma and companion cells, and everything is well preserved, the vascular cambium. So in many times when I can't see something in the bark, I take a razor blade and make a freehand section, and at least to be able to take some photographs and show some structures that I wasn't able to show with my slide. And many barks have a problem that it's ubiquitous, like when you're studying barks, that you have very soft tissues combined with very hard tissues. So if you see this image before, you have non-lignified cells, the sieve elements and the parenchyma cells of the ray and axial parenchyma, and you have very stiff fibers. So usually if you don't do some treatment, what happens is then when the knife comes, it crashes the whole structure and makes, the cambium detaches and it can fold like Alan showed in the beginning. And so we generally try to soften the material so that you get a more homogeneous structure to section. HF has been used for a long time in anatomical research. You put the material for a couple of days in HF. It's very dangerous, so I do not recommend it here. I include it because it is very used, but you need to make sure that you have everything in plastic because it's corrosive for glass. It has to be inside of the fume hood, but even the fume hood gets corroded by HF vapor, like the entire glass can get damaged. So generally we do not use anymore, but we have other methods that we can use. For example, ethylene diamine is excellent. You can use 4 or 10% and for 2 or 4 to 4 days, and it can be reused even after the HF gets dark. It's usually yellow, a light yellow, but it can be used again. It's all the bignoniaceae that I have section, I have used ethylene diamine. But for some plant families, it's not adequate. For example, for Malpighiaceae that I also study in my lab, in a couple of hours, the ethylene diamine makes the cambium detaches from the wood. So keep that in mind. That is not good for any material, but it's very useful for most of them. It is also common to boil in water and glycerin. You can do that in a pressure cook or just without it. It just takes longer, but it also tends to make the material more homogeneous. And in the lab that I shared with Teresa Terrazas, they use glycerin with alcohol and water, and also with excellent results. It's 1-1-1. And it can, there are many advantages. You can leave it for an indeterminate amount of time, and it makes the material really easier to section. I remember Teresa mentioning that they kept some oaks in GA, and they had excellent results after leaving it for a long time in the solution. And so in these cases, after you soften, if the softening was necessary, if the material is already very soft, I do not recommend using it. But in case you need it, the second step would be embedding. Again, if you need embedding, of course, you can always do it without it. And generally, also the sample size matters. In this case, large samples, when you want to have an overall structure image, polyethylene glycol is my preferred method, with PEG 1500 is what we use, because it's solid in room temperature. And if you have a smaller sample, but still able to see like the overall image of the phloem, you can use paraffin, a Carlquist, late Carlquist that we lost recently, used paraffin a lot to study bark with excellent results. And if you want the very fine details, I will show slides later. And then metacrylate is the best. Hystoresin is the best method if you really want to see the details of the bark. But then it's not very permeable, so you need to have very small samples, lower than 4 cubic millimeters. And of course, depending on the method of embedding, you will have a different microtome used. I will show that in more detail. So I will show first the method that I have been using a lot, that is from Rupp from 1964, that uses polyethylene glycol. This is the entire reference that I can send if someone needs it. But it's quite easy to do. So usually what we do, and why is PEG good? Because it's water soluble. So it usually doesn't make the material stiffer, because some embedding methods can make it stiffer, like paraffin, because you have to dehydrate and use butanol, and it makes the material sometimes even harder to section. And there are 2 methods that you can use to embed in PEG. Either you do it step by step, like I will show now, or you can use the original method. In step by step, basically you keep the sample immersed in PEG. You can take directly from your jar, or after you soften and put in the jar with PEG here in 20%, and it has to be inside of a paraffin oven in 60 degrees Celsius. I don't know why I put a percentage sign here. It was supposed to be a Celsius sign. And then you increase little by little every day. You put it from 20 to 40 to 60 to 80 and 90. Then you go slower in the end, up to the moment that it's 100% in polyethylene glycol. And then you can open the lid, because what you want is because there's water in it, you want the water to evaporate. And in this step, we usually put it in a vacuum chamber. So we make sure that the material really entered. I had colleagues working with monocots that they used to boil in a PEG 100%. It also removes air if you boil. And recently in botany, I think it was last year's botany conference, some person recommended using a little bit of PEG 4000. If you are in a moist area, it makes it more solid in the last step, because sometimes polyethylene glycol is it can take the humidity of the air and puts in the sample in the block, and it makes harder to section. Or we can use as the original recipe suggests. So from Roop, what he says is to put the sample in a very large jar, the largest jar you have. You cover directly with 100% PEG, directly from the field. And you just you can mark with a pen, so you know where the 100% PEG was. And you just fill the jar up with water. And then you shake, make sure that the solution is homogeneous. And then you can open the lid, put it inside of the paraffin oven again, or any oven at 60 degrees Celsius. Again, I have the percentage. and then you leave the water evaporating little by little. It should take a week or a week and a half to have the whole water evaporated. And if it's too warm, it will go faster. So that's not good. You want it to be slow, because you want to make sure that the water is coming out of the material and going into the air little by little. But I have used both methods, including little by little and including like Roop suggested, and the results were equal. So this is easier, and also use less material. And then you make an origami paper box, and you can put your sample inside of it and just fill it up with a PEG 1500, and it will dry, and it will have something like this. And of course, remember putting like what it is, because you can easily forget. And the amazing thing about it that you can keep it for an indeterminate amount of time. So when I was moving from Sao Paulo, then to Washington, D.C., and then from Washington, D.C. to Mexico, I was traveling with all my blocks, and I still section them after 6 years, and it's just as good. So it can be kept this way, and it's much easier than like moving things in alcohol, which is also, you have to lie basically to the air company, because nobody wants to have alcohol in the luggage. And if you can keep in silica gel, because again, it's water, it has affinity with water, so you can take water from the environment. So you put in silica gel, or you can put in the fridge. And then it has to be sectioned in a sliding microtome. And here again, I can't stress enough how important it is to have a very sharp knife. This is the most important thing. And of course, also the inclination, the angle that you use for the knife is very important. So if you're sectioning with this angle, it means that, well, here's a longitudinal section, so it's not so difficult to section. But if it's a hard plant to section, you want to increase the angle, and then it will make easier to section. And also, if it's too stiff, you want to section thinner slides, because then it's easier to make the knife go through 15 micrometers than 20. And then I will move to this technique that we have been using a lot, that was created by Antonio Barbosa, that is here in the image with Adriana Costa and a younger self. And he is an amazing technician from the Institute of Technological Researches of Sao Paulo. Many of us here in the audience probably know him in person. He was in Madison, Wisconsin, working at the FPL for a couple of years with Veronica Angelossi. And he's really amazing, because his mind is completely open to imagining things. So for some time, he was embedding plants in soap, and he was also working, and he is always trying new things. And he invented a resin that in Portuguese we call it resininha do seu Antônio, that is basically like what he created, and I will show now. So basically what you do is you take styrofoam, and you take pieces of styrofoam and you dilute directly in xylene or butyl acetate. and probably HistoClear works too. We have to ask then some people that have been using that now, but you dissolve the pieces of packaging that you receive, you dissolve in xylene or butyl acetate that is less toxic, and you will have a resin in the end. Then on top of the sample, you can brush, you coat the sample with this brush, and then it creates a sort of a pellicle or something on top of it that will hold the structure together. So what happens, the problem with barks, remember, is that the fibers get to one side and the sieve elements to the other. And if you have this coat on top, it will make sure the whole structure is stick together. Sometimes we combine this with an adhesive tape afterwards, we put on top of it. After it's dry, you have to let it dry. We sometimes even take a fan, or we use like a piece of paper, or we can put a fan on the side to make it dry faster, or take a book because then you have to wait. But again, patience is key, because if you lose time here, you save time later, because if the images are good, then you won't lose time looking, where's my sieve element? So if you put also an adhesive tape on top of it, you can use as another thing, and you can take with the adhesive tape, and you can even put in a line and wait to mount later. But it can also be done without the adhesive tape. So you take with a brush and you take the sample and put directly in water. The moment you put in water, the sample, it already dissolves the polyethylene glycol that is water soluble, but the resin stays. And the advantage of having the resin here is that you can stain, if you are bleaching, because you can bleach in some cases. Be careful with bleaching, because when you put chloride, you remove many of the cell contents. So if you want to check in case of barks where the sieve elements do not collapse, you will need the contents to make sure that which part of your phloem is conducting. Because if you bleach everything, then everything will be empty, the companion cells, and you cannot make sure up to which point the phloem is conducting. And of course, in the last step is the most complicated, because you dehydrate and you have the resin all along, and you have also the adhesive tape in this case. But the moment you put xylene or butyl acetate or HistoClear, the resin will dissolve. And in this moment, you can have breaks of the structure, because then the structure is free again. But you have saved all the other steps. You have saved your sample by having the coat. Here, just showing bleach, as I put here this exclamation point, just to make sure that if you are bleaching, do not bleach all the samples. You can bleach half of them to have more beautiful samples, and the other half you leave them intact, so you can check the sieve tubes. With double stain, I will talk about some double staining methods. You dehydrate, going through several ethanolic in an ethanolic series, and we use butyl acetate. But here you can see the samples, they are within the resin. You can see that they're square and they are saved in this way. And it really works well. So here, just to show up some images that we section this way, the good thing is that you can get sections of entire stems if the stems are not too large, like in the case of lianas. But you can also have a good image of details here in Ficus pumila. And here another example. This is a very hard wood, so this was softened with polyethylene glycol. So this is one of the many iron woods that we have across the globe. And here showing that you can see some details in Leucina. You can see sieve elements, you can see many cells. But of course, when you compare a section of the same plant done with PEG and done with metacrylate is superior. So I'll talk about a little bit about that following. So here you can see that although you can see the arrangement of the sieve elements and everything, you lose a lot of the details that you can more clearly see with historesin. But you can see that for wood, it's actually better to have PEG, because you have a thicker section, then you can see the lines of parenchyma and everything. But metacrylate has many advantages. The only disadvantages is that it needs to be in a very small block. So it's very good for details, but hardly, most of the times you cannot have an overall image, but you can, here you can clearly see that you see the sieve elements, the companion cells, parenchyma cells, the vascular cambium. Here in sieve elements with nacreous walls, you can see them perfectly. Here you can see the fibers differentiating next to the cambium. So it's the preferred method when you want to describe the phloem. You only have to follow the key instructions. It always comes with the instructions. The difference that we have, and that I have been seeing people doing, is then it suggests a very short period in the pre-embedding and the embedding solution. And we just recommend making it longer. So sometimes even for a month in each solution, and then you have a very good embedding. And of course, with hysteresin and paraffin, you section in a rotary microtome. And here I'll speak about some staining techniques. And I use in my lab preferably Safra Blau, which is a German invention, makes our lives much easier. It can be done for overall tissues embedded either in paraffin or PEG. And what we do is a solution. We make a solution of Astra Blue, 1 gram of Astra Blue dissolved in 50% alcohol, and 1 gram of safranin dissolved in 50% alcohol. And then we make in a jar of 90 mils of Astra Blue and 10 mils of safranin, and you get the solution mixed. And the good thing is that the double staining does automatically. You just put some drops, and in 15 minutes, 10 to 15 minutes, you have the section stained, and we double stained. And you can see here, for example, it stains perfectly. And in only 15 minutes. So it's really also a very good option for classes, because you don't want to spend so much time in the classes staining. So you can use Safra Blau. Astra Blue, you have to know very well which Astra Blue you're buying. So I got Alan has a good source of Astra Blue. They can from Germany. I know that they have some problems now. They are not sending to Europe. I don't know why, but maybe that you can find a way around it. But their Astra Blue is the best, because in the United States, the one that I bought didn't work. So I don't know. And also works for other types of tissues, just to show a monocot, and it works perfectly well for them too. Here's a palm stem.
Marcelo, you have 5 minutes left.
Okay, thank you. Here, a palm tree that you can see also sectioned with the Antonio's technique. But again, this with metacrylate, usually for metacrylate for hysteresin, we use toluidine blue. That it's also just a couple of drops, and also it has metachromacy, so it will have whatever has only primary wall will stain different from things with secondary wall, so it's also quite good. Fast green and safranin work just as well, but you need to leave in safranin for longer, because fast green is really strong, so you can wash away the safranin. But here, a photo from Teresa Terrazas, they use usually fast green, and it works perfectly well too. And one that I want to speak quickly is about a Lacmoid that is here in this publication. You can find the recipe and how to do it, but it has an advantage for phloem. And although the overall is not so beautiful, the staining, because it gets a little brownish because of the ferric chloride, it has this amazing advantage that Lacmoid Resourcing Blue has affinity for callose. So you will much easier locate your sieve cell or sieve tube element, because it will have the callose in the sieve plate. Here you see in a longitudinal section, you can see how the callose gets stained, and you can even detect very clearly the pores in the sieve plate. And also in the sieve areas, you can see here in this Quercus alba, you can see the sieve areas with the pores. And here in this Castagna, you can see how the Resourcing Bloom gets perfectly well in each pore. So it's really, Everett used this staining for all his sections. I have also used, and it's amazing really if you need a easier way to detect callose and sieve elements. And you can also use aniline blue, but I won't have time to go through it now. As I said in this book by Johannes Lische, and I have summarized all these methods. You can search there, and and I can also give you the PDF. I think the book's available online free of charge. Some open questions and challenges like, can we find a substitute for celluloidine? I think if someone is using that, I would love to hear about it, if it's working. Just to show here another photo of Tilia americana basswood, where you can clearly see that it was really good, even with the cambium is active. And does micro CT has any role in bark studies? Is a question I think it doesn't, answering myself, because we need fine detail, and sometimes I can't see that in micro CT. And also one thing that it would be lovely is to build a database like we have inside Wood for barks. That's one thing that we have to ask Elizabeth Wheeler to this initiative. It would be really great to have a database for that. And I thank you very much. And I hope everything was more clear than not. But feel free to reach out if you have any question.
Thanks, Marcelo, for the huge amount of information you provided and the details of it. I really enjoy your talk. And I see the chat is live. Don't forget to write down your questions. The questions will be answered at the end of the workshop. And we now have basically 20 minutes for a break. And we will meet here again at the hour. So in 20 minutes, wherever you are, it's 20 minutes. Thank you.
Good morning or good afternoon, depending on where you are. I would like to start by thanking Alan, Ilana, and Angela, and the rest of the organizers for the invitation. I'm very excited about being here. It's very cool to be among people interested in bark. It's unusual, so it's very nice. And I'm very happy to have the opportunity to share with you the research that we have been carrying out for the last 10 years or so. And all of us have noticed the great diversity of bark, right? It's just a matter, as Alan was saying, of going out to our gardens. If we go to the park near our houses, we can see all kinds of barks, different textures, different colors. There are barks that lose chunks like this Melaleuca from Australia here, or there are other barks that peel like this Proteaceae here or this Persera here. And despite the fact that bark is super conspicuous, as Alan was saying in the beginning, and despite the fact that it's very diverse, we know very little about bark ecology and bark evolution, especially in comparison with wood. And with this talk, I would like to make the point that one of the factors that has hindered our understanding of bark ecology has been the way in which it has been studied. Traditionally, bark functional ecology has focused on only one of the functions that bark carries out for plants, that is, the protection it provides against fire. And I hope that by the end of this talk, you will agree with me that taking into account all the other functions of bark is very important to understand this remarkable morphological and anatomical diversity we observe in bark. And as has been mentioned before, bark refers to all the tissues external to the vascular cambium. And we can see in this cross section, can you see my pointer? Yes, yep. Thank you.
Yes,
thank you. And as we can see in this cross section of this Malvaceae, this Brachychiton from Australia, bark can be very massive. This is a twig, and you can see that in comparison with wood, bark can be very, very abundant. And in the inside, we have seen many beautiful slides before my presentation. And we can see that it's also very, very complex from a structural point of view. And I don't want to spend too much time on this slide. Previous speakers have explained some of the structure much more nicely than I would be able to. But with this slide, I would like to highlight that from a functional point of view, we can divide bark into two major regions. We have the outer bark that's composed mainly of, well, absolutely, totally of dead tissue. And we also have the inner bark, which is made up of mostly living tissue. And in the case of these species, the inner bark would comprise all these tissues, and the outer bark would comprise the phloem. But as Simcha was mentioning earlier, there are some species that have Rhytidone. So many species outer bark is made out of just Rhytidone. And I want to highlight that this this typology, this reference to outer and inner bark, the one I'm using, differs from that from the International Association of Wood Anatomists. And this is because we are highlighting here a functional perspective, not an ontogenetic one. So don't get confused, please. I'm using the terms slightly differently, but for that reason. And given that bark is derived actually from two different meristems. It's derived from the vascular cambium that would be here, and it's also derived from the phylogen, the meristem, the cork cambium that produces phylum to the outside and phyloderm to the inside. Given that we have these two different ontogenetic origins for bark, and also given that these two regions, outer bark and inner bark, have different functions, as we will see a little bit later, we see a very different combinations of these two regions, inner bark and outer bark, very different proportions. Here we have cross sections of savanna species from Australia. These are twigs again. And we can see that inner bark and outer bark can vary widely in proportions. For example, this cross section here of this species, this is the Brachychiton that we were looking at earlier. We can see that outer bark is just this very thin layer here, and inner bark is very abundant. In contrast, we see in this Apocynaceae here that outer bark is very thick in comparison with inner bark, which is this whitish ring around the wood. And we see that in this plot here, we have around 100 species arranged by their total bark thickness. And we can see that plants species with very similar total bark thickness can have very different compositions of inner bark, which is shown in gray, and outer bark, which is shown in black. So we can see, for example, species with very similar total bark thickness can have very different compositions of these two tissues. And this must have very different or very important implications from a functional perspective, because imagine you have a bark which is composed mostly of dead tissue in comparison with one that has mostly living tissue. And we have here very interesting extremes. These two species, Alocasarina and this eucalypt, both from Australia, they were growing side by side in Tasmania. They had very similar total bark thickness, but look at the very different proportion of outer bark and inner bark. It couldn't be more extreme. Alocasarina had mostly outer bark and very little inner bark. And in contrast, Eucalyptus punctata had the opposite trend. And we can see that the total bark is very similar. So we see very high diversity in bark, in the amount of bark, in the structure it has. And despite this fact, functional ecology of bark has mainly been explained by fire regime. And this perspective has been the norm in the literature. And in that sense, total bark thickness has been the central trait. It's a relatively easy trait to measure. It's the most measured trait in bark, and it's highly variable. It can go from less than 1 millimeter to 30 centimeters in the cork oak. And we know that bark protects the cambium and the phloem from necrosis when there's a fire. And also more recently, it has been shown that bark protects the wood from deformation and embolism, which are additional factors that can cause death after a fire. So thick bark has been regarded as a paradigmatic adaptation to fire. And it has been explained through a fire regime globally, traditionally. And we will call this the fire-only perspective or fire-only paradigm. And following this paradigm, we can see that savanna species, and I apologize for the lack of scale on my pictures, but believe me that these cross sections on the left have very thick bark. They are species from the Brazilian Cerrado, the Brazilian savanna, very frequently burnt area. And yes, this is congruent with the fire-only perspective. Outer bark is here, inner bark is here, total bark is very thick. This species shows the same pattern. And in comparison with rainforests, we have very, very thin barks in comparison. So yes, the paradigm seems to work when we use these examples. But what happens when we look into these two other species? Again, I apologize for the lack of scales, but believe me, this species on the left is has very thick bark, and the one to the right has very thin bark. So you would say, yeah, well, following the paradigm, the fire-only paradigm or the fire-only perspective, this species must come from a savanna or a fire-prone environment. And this one to the right has to come from a fire-free area, right? Well, let me tell you, I hope you will be surprised when I say that both come from a non-fire-prone tropical forest. So we see that things are not that simple as to infer that plants with thick bark grow in environments subjected to fire, and plants with thin bark grow in environments without fire as a selective agent. And to further explore this question, I want to share with you some of the results of one paper in which we sampled around, well, actually we sampled exactly 640 species from 18 sites in different parts of the world. We have half of the sites in fire-prone areas and the other half in non-fire-prone sites. And we measured bark thickness to examine how this trait varied across sites. And we also measured stem diameter because we know that bark thickness follows stem diameter. So we have to take into account the size of a tree when we make these comparisons to make them fair, to make them logical and yeah, fair from a statistical perspective. And to take size into account, we calculated residuals for each species. So here in this plot, we have different points representing different species. We have this positive, this trend, no, in which bark thickness increases with stem diameter. And we calculated the distance from each point to the mean regression line. And we see that some species will have negative residuals, meaning that they have thinner bark than expected, given the size of the stem. And we have plants that have positive residuals, that have thicker bark than expected, given their size. So we were interested exactly on these differences here. And what did we see? Well, we found that, well, first of all, we built a box plot like this one here, and you can see that, well, we have this line here that shows residuals with a value of 0, that is species on this region of the plot would mean that they have the thickness of the bark is expected given their size. And we have the different sites, the 18 sites that we sampled here. And in red, I represent the sites that are subjected to fire naturally. And in black, I'm presenting the fire-free areas. And as we can see, yes, this is congruent with the fire-only perspective. Savannas have barks that are thicker than expected. That is, they have positive residuals. And yes, this is congruent, as I was saying, with the fire-only perspective. Bark protects against fire. But at these same sites, we also have species that have thin bark that is thinner than expected. So fire-prone sites, the important message here is that, of course, there's thick bark, but there's also thin bark. And also, something that I want to highlight here is that non-fire-prone sites, like this seasonally dry tropical forest that does not burn naturally. I mean, it can burn if you light a cigarette and drop it on the ground, but it will not burn naturally, or at least it will not burn as with such a frequency as to represent a selective factor in the lives or in the evolution of this species at this site. So as I was saying, in this dry tropical forest, species can have very thick bark as well, even in the absence of fire. So the other important message here is that non-fire prone sites also have thick bark. So why do we observe so much variation within communities? And we have thick barks also in areas that do not burn, thin barks in areas that burn. So what's going on? What's happening? What's the problem if fire regime is the explanation for bark variation, right? Well, the rest of the talk, I would like to make the point that there are different ecological strategies within communities. If we focus on bark, we will see different things. Plants will be optimizing different functions. And the main message will be that bark diversity and coexistence can be explained if we take into account these other different functions. And before moving to the discussion of the different functions that bark carries out for plants, I would like to open a very short parenthesis to talk about multifunctionality in biological structures. And as biologists, we know that most biological structures carry out multiple functions, right? And I have here an example from zoology to have something about animals in this symposium. And we're going to be talking about the femur. The femur is the longest bone in our body, and it has different functions, mainly four functions. It provides mechanical support as part of the skeleton. It's also a very important region for muscle attachment. And it's also hollowed, right? It's hollow and it has the bone marrow inside, and the tissue inside the femur and many other long bones allows the production of blood cells. And also the femur is involved in the regulation of calcium balance. This bone releases calcium for metabolic processes in our bodies. And inevitably, when a biological structure carries out more than one function, which is the case most of the time, trade-offs will emerge among these functions. And let's focus on some of the trade-offs that emerge in the femur. So in the case of the femur, a hollow area in the center provides room for the marrow for blood cell production, but reduces mechanical support in comparison with a solid cylinder. A solid cylinder would be much better than a hollow cylinder for mechanical purposes. So there's a trade-off between the function of mechanical support and blood cell production. Also, the release of calcium, that's another function as we were discussing, regulates calcium balance, but reduces mechanical stiffness as well. So there's again a trade-off between mechanical support and the calcium balance. So if we wanted to understand the shape of the femur, focusing only on mechanical support, it would be impossible. It would be super strange to see that the femur is hollow. It wouldn't make any sense from an evolutionary perspective. But focusing on mechanical support is only giving us a partial picture of the many selective pressures that have shaped the femur. So I wanted to highlight with this that we need a multifunctional perspective to understand biological structures. And this multifunctional perspective is unfortunately not very common in botany. It's very common in zoology, but fortunately we have good examples in botany that we can share now. For example, wood. Wood is a very good example of a multifunctional tissue. Wood carries out several functions for the plant, such as water conductance, mechanical strength. It also stores water and other compounds. And using a multifunctional perspective, we have started to understand the diversity of wood, but we have to take into account the different things it carries out for plants. So this multifunctional perspective is essential. And I want to highlight the point here that this multifunctional perspective is essential to understand bark diversity as well. So we will be talking the rest of the talk about the different functions that bark carries out for plants. And as we were saying, bark protects from fire. Of course, it protects from fire. I'm not saying that it doesn't. I'm just saying that it's not the only function it carries out. And bark also protects from desiccation. It protects from temperature. Here we have a very neat example of that. These are eucalypts that are called ghost eucalypts from the center of Australia. They're bright white, and it seems that this color of the outer bark protects from temperature elevation. And bark also protects from pathogens, from herbivores. It's the first tissue that responds when there's a wound. Simha talked about this. I'm not going to talk much more about this. And as I was saying, of course, bark protects from fire. And we have recently, we as a community, we have recently discussed that it's not total bark thickness, the trait that provides protection against fire. It seems that it is outer bark thickness, the trait that's important, that's central for this protection. And we can see that here in this comparison of the trunk and root of these species from the Brazilian savanna from the Cerrado. So this species, we can see that in the trunk, it has very, very abundant outer bark. And look at what happens in the root. The root has very thin outer bark. So it makes sense, right, to have outer bark in the area that's exposed to fire and to have much less of that tissue in the area that's not subjected to fire. And another empirical evidence of the role of outer bark is this one here. So these are the sites, the 18 sites that I mentioned earlier, sites that burn. The ones that have a little fire above are the ones that burn. These ones do not burn. And we can see that the inner bark proportion increases in areas that do not burn naturally. What does this mean? Well, areas such as savannas that are very frequently burnt, or Mediterranean woodlands, or sclerophyll forests, all of these sites experience frequent fire. These sites will have a very high proportion of outer bark in comparison with inner bark, as you can see here. So these areas that do not burn, the barks here usually have very thin outer bark. So there's a trade-off between fire protection that's provided by outer bark and the amount of inner bark in a bark. And another function of bark that we need to always take into account has to do with the photosynthetic activity of the living region. Of the living region of the bark. And photosynthesis in bark recaptures respiration CO2. And there are studies out there that show that, for example, 11% of the wood carbon is derived from bark activity in eucalypt twigs. There are other studies showing that this number can change, but we need to have more information on the contribution of bark to the carbon balance of plants. And we have measured how photosynthetic bark, the probability of observing photosynthesis in bark, decreases as outer bark thickness increases. And this makes sense, right? Because outer bark is a layer that can impede the penetration of light. So the thicker it gets, the less likely it will be that light will be able to penetrate to allow photosynthesis in bark. So again, there's another trade-off between fire protection and photosynthesis. And this is something I wanted to highlight, because some species in fire-prone sites, such as savannas, these species seem to optimize photosynthesis at the expense of fire protection. So they have very thin outer bark, but they carry out photosynthesis. So there must be a compensation there in terms of the performance of the plant that's selected for. So these plants have very thin outer bark, but they can carry out photosynthesis. So there must be something good about it that's selected by natural selection. And another function of bark has to do with storage. We have recently published a paper on this, in which we show that in terms of concentration of soluble sugars, we can see that bark, which is the whitish plot here, this is wood. So we see that the concentration of soluble sugars is higher in bark in comparison with wood across all organs. So bark is more important in terms of concentration for these compounds. And when we look at starch, we see that bark is more or less, well, it's very similar to wood at the level of twigs, at the level of trunks. The situation is reversed at the level of roots. In roots, wood has a higher concentration of starch, which makes sense, right? Because we usually associate roots as storage organs, right? And with this slide, I only wanted to highlight that bark, inner living bark, is carrying out very important storage functions for the plant, not only of carbohydrates, but also of water. We see here, this is these are two plots from another publication in which we measured the inner bark thickness, taking into account the size of the plant. That's why it's residual here. So we can see that the amount of inner bark decreases with the amount of precipitation during the dry season. So that means that this seems to suggest that inner bark is carrying out storage for plants. It's storing water, especially in seasonal areas. And we see also that inner bark increases slightly, but it increases with the temperature. So hotter areas have more abundant inner bark. So again, there can be a trade-off between the storage function of bark that's carried out by inner bark and the protection that bark can confer to plants through outer bark. And another function I wanted to highlight is mechanical support. Bark also provides mechanical support for plants. And here we have a plot that shows the contribution of bark to mechanical support. Here is distance from the stem tip. So you would have the tip here, and this would be older regions of the stem. And we can see that bark, this work was carried out on Bursera. It's a tropical tree, the gumbo limbo, for those that have been to Florida.
Julieta, you have 5 minutes. Okay.
Fantastic, thank you. And so we can see that the contribution of bark can be relatively high, 40% at the tip of the twigs. And we would never think of bark as a mechanical support tissue, right? We always think of wood as the main mechanical support tissue, but bark contributes, especially in the younger portions of stems. And these younger portions of stems sometimes carry the fruits. So it's an important tissue providing mechanical support. Of course, this support decreases as the branch gets older, but it's still relatively high in this group of trees. It's around 20%. And bark also co-varies with wood. So it's not only that bark is, as a traditional fire-only perspective has postulated, bark is not only a passive layer, a passive skin around trees. No, it's a region that co-varies with wood very strongly. For example, we have here the covariation in terms of density. There's always very strong covariation between the wood and the inner bark. And the inner bark is always less dense than wood, but still it's the covariation is very, very strong. You wouldn't be surprised, maybe, because you know that these two regions, the secondary phloem and the wood, share an ontogenetic origin. But for ecologists, sometimes this ontogenetic link is not very well known, and they are surprised by this kind of covariation. No? But this suggests not only an ontogenetic link, but also a functional link between these two tissues. And bark also co-varies with leaves. And this is something that has been poorly explored, but it seems that thicker bark is associated with larger leaves. And there's some economic spectra involved in this covariation. I'm not going to discuss this today, but the main message here is that bark is very finely tuned with other plant parts. So we need to go beyond the fire-only perspective and beyond our perception of bark as just the skin. And the main messages I would like to highlight for you is that the fire-only perspective will only provide an incomplete picture of the causes of bark diversity, and that other functions need to be taken into account to really understand the amazing diversity of bark, both externally and internally in terms of anatomical structure. And there are very diverse ecological strategies in bark, and we're starting to understand them. it seems that different species optimize different functions, and there must be selective reasons for that. And we're starting to understand what's going on, the kind of trade-offs and synergies between these functions. And these diverse ecological strategies are also observed within fire-prone communities. So species are doing different things within fire-prone sites. And bark seems also functionally coordinated with other plant parts. And the organizers asked us to bring some open questions in the field we work in. And I brought these 3 questions. There are many more questions in bark functional ecology. It's a very open area, especially, I'm saying this to students because there's a lot of things to discover in this area. So if you're interested, please contact me. And one of the questions, I think it's very important, has to do with how does inner bark, phloem, and other living tissues, not only the phloem, but also the other tissues that are carrying out storage and other important functions, how do these tissues scale with other traits, for example, with leaf area? There are a few studies focusing on this, but we still don't know how this scaling occurs in plants. We also need to know what the role of inner bark is in the water balance of plants. There are, again, a few studies out there, but we still need to understand what's going on and how bark contributes to this important function of plants. And there's a very basic question that we still don't have an answer to, and it is how much bark is there in a tree. So we understand how much bark there is in trunks because it's of economic importance to know how much bark there is and how much wood there is for, as I was saying, for economic purposes, but we don't know how much bark there is in crowns, and we really need this information. And it's very important to know how this amount varies across species and across environments. And I think that's it. I would like to thank you all for your attention, and well, I'm looking forward to the discussion. Thanks.
Thank you, Julieta, for a very nice presentation. Now we want to welcome Alexey Oskolsky with his lecture on topic of relationships between bark macroscopic appearance and its microscopic structure.
Okay, listen for me just a moment. Sorry. Okay. So hi everyone. So thank you. Thank you very much for invitation to organizers for invitation me to present my talk in this symposium. And so I am so happy to listen to such an interesting talks, and I would like to share some my suggestions about the relationships between bark appearance and bark anatomy. So my talk is based in essential part on our joint my collaboration with Ekaterina Kotina and my discussions with her, and in systematic and evolutionary and ecological bark anatomy in various plant groups. So,
can you please put your presentation in presentation mode?
Sorry, is it not in presentation mode?
No.
Sorry, let me. Now it's okay.
Wait, no.
Is it no? Strange. Sorry.
There is a button показ слайдов.
Sorry.
Alex, it's in the bottom. You have it's fine. Yeah, there you go.
It's okay now.
It's okay now.
No? Okay, thank you. Sorry. Okay. So you know that the appearance, macroscopic appearance of bark is very diverse, and microscopic structure is also very diverse. But actually, this is two different realms, and we know quite a few connections in between. And I can say that macroscopic structure is even much poorer explored than its anatomy. And there is not commonly accepted terminology for a description of bark macrostructure. And of course, there are very important publications of Mikhail Trockenbrod and Leo Unica. They propose elaborated, quite detailed terminology for description of bark structure, bark appearance. So unfortunately, Leo Unica's work is not completed. It was not included in recommendation of International Association of Wood Anatomists for bark descriptions. But what is the anatomical terminology is much better elaborated. But how to connect it with how to understand the relationships between macrostructure and microstructure. So there are quite few studies, few works, which are focused on this matter. I think the most important works are the several publications of, so it's Thomas Whitmore in 1962 and 1964. They are focused mostly on bark morphology and bark anatomy of the family Dipterocarpaceae. And he also studied some, in detail, the bark structure of some temperate trees. And some important ideas of Whitmore are considered later. And in my plan, I would like to propose some classification of types of bark macrostructure related to its anatomy. So firstly, I discuss this classification, then consider the variation of plant anatomy of different macro types of barks, and then briefly discuss the shifts in age shifts between different types of barks. So, and first of all, so for the, let's start with very simple things. Let's forget the bark anatomy for the time being. So, bark is a complex of tissues to the outside of vascular cambium. And usually the entire bark is affected by dilatation. But bark is a protective structure. It must be covered by continuous protective layer. Then inner region of bark is not collapsed, is poorly dilated. Outer not conducting region is usually heavily dilated, it's collapsed. And taking it in mind, we can see that the bark can have different ability for dilatation. Some barks, in some barks, dilatation is quite limited. It is just cell expansion and maybe the cell sclerification, and some only a few divisions, cell divisions. In other barks, we can see the conspicuous dilatation. And for instance, the presence of dilatation meristems, or the meristematic activity for the and for the dilatation. We can see, take these two conditions, inconspicuous or conspicuous dilatation. Then we can see that collapsed tissue can either persist on the bark or can shed off. And in the bark, we can some barks, we can see the regular separation layers. They provide regular shedding of the outer regions of bark. All of these separation layers are lacks. So then we have combining. We can have we can consider 4 combinations of these two conditions. So if the bark can have conspicuous dilatation, at least in outer region, or inconspicuous, and it can have the separation layers, regular shedding of the outer parts of bark, or these separation layers absent. So that's we can recognize 4 combinations. But what is interesting that these just speculative combinations can be very we can recognize these combinations visually. So, and I propose the terms for this combination. It's stretched bark for conspicuous dilatation without separation layers, exfoliating bark with separation layers and conspicuous dilatation. And in inconspicuous dilatation, we can see the fissured bark without separation layer or scaly bark. So that's, and we can see this visually, macroscopically. We can easily distinguish between these 4 types of bark. So, and now, let's consider some anatomical features of each of these types of bark. And we can start with stretched bark. So, as an example, we can find it in, it is baobab, Adansonia digitata. It's Triphylia or the Betula. And usually they have smooth surface and eye-like markings on the surface. What about the anatomy? So that usually this the protective layer in these barks, it is periderm, but periderm is this single periderm without retinol, and usually it is quite thin. It's able to stretch, and but these barks have prominent pseudocortex. So the pseudocortex, it's a term and concept proposed by Whitmer. It's a name for parenchymatous zone of mixed origin. It's derived from cortex, pheloderm, and maybe the parenchyma of secondary phloem, but it's impossible to distinguish between the source of this parenchyma. Usually also that these barks have dilatation meristems in different layers of this bark. They can be in pseudocortex and can be also in phloem. So then, next type is exfoliating a bark. It is they have also smooth surface, but with more or less distinctive scars of falling portions of thin portions of bark of outer region. But these regular scars suggest that this is the separation is a result of presence of separation layers, some separation structures. But what we can see, as an example, we can find consider it in Platanus. They also have single periderm. They usually have more or less distinctive pseudocortex, and they have dilatation meristems, at least in the plants about which I found the literature. But they have the SS separation layer is probably the result. This is the layers in phylem, thin-walled layers in stratified phylem. Then Fischer bark. This is the bark with inconspicuous dilatation, limited dilatation, and the lack of separation layers. So this it has rough structure, rough surface, usually with vertical fissured and separated by ridge, and without evident threads of detaching of some portions of bark. We can see this this structure. Actually, this is the textbook example of bark. This is the barks with thick rhizodome, and the rhizodome is the protection layer, including the periderms, many periderms, and collapsed phloem. This is the example of this bark from Quercus petraea from the paper. So these barks have no pseudocortex, and I didn't find the information about dilatation meristems in these barks. So, but this is one of the possible options. But we can find such the appearance of bark in plants having no periderms. Such condition present in some woody plants which have interaxillary phloem. They have phloem inside the phloem strands are formed to inside of cambium, and the cambium forms the so-called secondary cortex to the outside. Actually, this secondary cortex works as a protective layer instead of periderm. And so this is the bark of Metallazia densa. This is the shrub of the family Asteraceae from South Africa, and we can see this. This is the secondary cortex. We can see the transformation of outer region of secondary cortex, the thickening of walls in this, but this is not the formation of periderm. This is just the transformation of cells of thickened of these initially parenchymatous cells. And we can see, there is no separation layers, and as a result, it forms the fissured bark also. So then, scaly bark. We can see that scaly bark have rough surface with prominent traits of detaching portions. And this detachment suggests that the presence of separation layer. But these the portions can be stringed, can have different shape. And this is some examples of scaly barks. Important is presence of separation layers. So, but how it works? It's interesting that usually in these barks, the protective layer is the sclerified secondary phloem. Collapsed secondary phloem is protective layer. But periderm, at least phylem, works as a separation layer. So you see, this is the bark of Buddleia. It contains thick bands, the bands of thick-walled elements with very thin layers of periderms. The periderms can be just uniseriate. And the function of these periderms is obviously, this is just the separation layer. And these strips consist of sclerified secondary phloem, solid sclerification. But the most plants with scaly barks have such organization. They, of course, they have no pseudocortex and no dilatation meristems. Okay, but this is not only in scaly barks. This is the Ekebergia capensis, quite big tree of the family Meliaceae, from the South Africa, and detailed structure shows that the separation layer in this tree is also the periderms, and outer region of bark is covered by collapsed phloem with abounded tannins. But periderms serve as the separation layers. This tree is also interesting by presence of so-called phloem proliferation tissue. It's this is also Whitmore's term concept. This is the bands of result of radial expansion of conducting phloem. You see these structures. This for us is quite common. We can know that phloem can dilatate, stretch in tangential direction, but we can see the cell divisions in phloem, and to increase, to expand the bark in radial direction. It is not common condition. Similar condition have been described by Margaret Chataway in some eucalyptus, but these eucalyptus species and Ekebergia both have scaly bark. Maybe it is also take part in formation of this pattern. And so I just want to show this phloem proliferation tissue, formation of radial files of parenchymatic cells in conducting, in not collapsed phloem. Okay, and in some plants, it is not very common. We can see the opposite functions. The protective layer is phylem, it's periderm, together with sclerified secondary phloem. And separation layer is probably non-sclerified. It is in grape, in which is vinifera. And probably similar condition there is in some roses, but it is not very common. So to summarize, we can see the following combinations of characters. We can see 4 major types of barks, and stretched barks and exfoliating barks are characterized by thin protective layer made of periderm and by presence of dilatation meristems and pseudocortex. And fissured barks can have different organization, but usually they have ritidom, but some barks have no periderms. And then we can find the fissure bark on the base of secondary cortex. So then scaly bark can be also of 2 types. In most common type, the collapsed phloem works as protective layer, and separation layer is periderm. Sometimes we can see the opposite function. The protective layer is periderm, and a separation layer is the parenchymatous zone of phloem. So, and it's interesting. Of course, these morphological or functional types are they are not. They can be changed with the age of tree. And so this I looked. I tried to find some trends in transformation of the bark with age, but commonly the bark, young bark, have a smooth surface, of course, and they mostly have some traits of stretched bark. But with the age, we can see the change to exfoliating bark, scaly bark, or from scaly. In some, for instance, in Casuarina, we can see the changes from scaly bark to fissured bark. So these are the shifts that I just observed in some trees. This is not complete diagrams, and its result is I observed about 20 species at our campus. So that's, of course, it must be, we can change, we need to change it, check it in more detail. But generally, of course, yarn stems, they have smooth bark without evident traits of dilatation. But then we can see different shifts. I think it's quite interesting field, and I will try to continue to generalize it. So, what about the conclusion?
Alexey, you have 5 more minutes.
Yes, I'm finished. So, we can recognize 5 types of bark. Each is a distribution by combinations of conspicuous or inconspicuous dilatation and presence of absence of separation layer, but they are quite distinctive by their morphological features. And so that stretched and exfoliating barks, unlike the featured and scaly barks, share the presence of pseudocortex and dilatation meristems. And in many scaly barks, the collapsed secondary phloem can function as protective layer, but periderms serve as a separation layer. So, and some open question. My first open question: what is the proposal classification? Of course, this is just the working hypothesis. What is wrong with these 4 types of bark? Then, of course, that it would be important to find the correlations between these types and anatomical feature. From other side, these 4 types is not enough to describe the diversity of macroscopic structure of bark, appearance of bark. More detailed consideration are important. Then, what are the ecological, evolutionary, and taxonomic implications of these bark types? And of course, there are many interesting details are suggested by these data. So we can see that the collapsed phloem serve as protection layer quite commonly, and how the microstructure of collapsed secondary phloem can be associated with this function. Then, what are detailed mechanisms of periderm stretching? Then, what is functional role of phloem proliferation tissue? It is very a few information about this phenomenon. And also, it would be interesting to pay attention on the barks lacking the pteroderms. For instance, the barks of some plants with cambial variants having intraxillary phloem, or the barks of some monocots. So, okay, these are my questions. Thank you for your attention. So, thank you.
Thank you, Alexei. Thank you very much for your detailed presentation, which already triggered some questions in the chat. So for all of you, keep using the chat to ask your questions if you have one. You can also raise your virtual hand and unmute yourself and ask any question you want anytime for the next half an hour or so. To kind of break the ice, I'm picking up from the chat a topic which appeared at least 3 times. I saw that. And I have a question for our first presenter, Simcha Levgiadun, and also for Julieta Rosso. For both of you, the topic raised in the chat was about anatomical variation in the bark related to the distance to the tip of the plant, both in terms of hormones and how they concentration change along the stem, and both that this will be for Levgiadun. And for Julieta, on the wood side, we know that many wood anatomical traits, they change according to the distance to the top of the plant. Is that true also for the bark? And how much do we know there? Maybe Simcha, you want to start?
Thank you very much. And this will be, of course, to thank for the conference or workshop or whatever it is, because on bark, it's really rare that people discuss about it. We know very little. And as I said, people usually studied cross sections and didn't look about the polarity and about the ontogeny too much. The fact that it's not easy to section the mature barks, because as Marcelo gave us an excellent explanation and demonstration about it. Some people, I think, when they started to make sections, they cursed someone and left the job, because it's really complicated and not all people are stubborn enough to go for this. And you know, people start as a project students or a master student or even as a PhD, and if something doesn't work, many people will say thank you, bye-bye, and I won't see you again. And it became less fashion to study anatomy in general, and people who don't know anatomy come to various conclusions about gene action and things like this, and gene expression, which are delusional. And that's not what I say about it. And I saw unbelievable things about it. The bark is flexible because it responds not less than the xylem to environmental changes and to an ontogeny. And so if you have the side of the trunk which is exposed directly to the sun or to wind with salt and sand at the coast or whatever reasons, it can differ from the bark in the same place in the trunk, which is located 10 centimeters or 20 centimeters away. It can be smooth or rough in the same cross section of the trunk or a big branch, or even a small branch. I showed a little bit. It's very common, and we don't know enough about it. I say it, I am sad about it, but it's not easy to get grants if you propose to study things like this. For instance, the National Science Foundation in Israel, you have 5% in the grant proposal about technical innovation. So if there is no technical innovation and you lose 5%, this is the end of the grant proposal. You will get a very good, not excellent, and the university will give you something like $3,000 because you got very good. And this is the end of the grant proposal. And of course, nobody now studies plant anatomy in Israel, which was a center. And the same is true in many places, because here and there I go abroad and give advice to molecular people, because I bothered to go into molecular issues for several years, which was my major thing, just to know the language and just to know the way of thinking. So there is a gradient from young to old. It will change according to growth condition, to exposure, to upper and lower sides, to insect attacks, to flooding, to various types of wounding, for instance, in mountains, rock falls, or avalanches. And it's not documented because bark is not as important economically as wood. We know about wood, I will say, 5,000 times more than what we know about the bark. If we go for Phaloderm, I don't recall any essay focused on Phaloderm. Maybe it's my bad memory, or maybe it's not so bad and it's reality. So it's an open arena for anyone who has a position in a place where he can study it. Because in many departments today, you cannot study it. And if you have a question, you have no one to ask, what do you think about something in the next room? Because there is no one in the next room. So this net can help many people. Yes. And such meetings like today can help many people because, as I said, you cannot go to the next corridor. And in my country, actually, it's almost impossible for many things in plant anatomy, except for Ilana. I have no one else to ask what's going on, because Fan passed away and Ella Verger passed away, and Tova Arzi, who taught me the basic, passed away, and Nili Lifshitz, who was my mentor, passed away, and so on and so on.
And that's great that we still have we have the QNet, so we can share with.
Yes, and I'm the living fossil. I know that I'm the living fossil in many things. So every day people ask me questions, and but if I don't know someone, it's very hard to ask someone, and it's becoming a global problem because my young generation is disappearing.
Okay, thank you, Simcha, for your thoughts. I would like to hear Julieta on this topic about anatomical variation along the stem, from the tip of the stem downwards.
Thank you, Alan. Yes, this is a very interesting question. And there are very drastic changes along the branches of trees, from the very tip, the very mature tissues, all the way to very mature tissues at the bottom of the tree, of course. So these changes, these anatomical changes, are associated with important functional changes. For example, density of the bark increases, and there are differences also in the storage function of bark along the stem. We still need to work on this a lot to understand in a better way how these functional changes occur in association with anatomical changes. But I have a student now that's been sectioning trees from the tips to the trunks, trying to understand how the amount of the different regions of bark change depending on the environment, on the size of the tree, and also depending on the phylogenetic lineage. So in summary, it's an open question. We know it changes, but we don't quite understand how the function and the anatomy are associated along the changes along the stem.
Julieta, do you know if it's hormone related?
I guess it is. I'm not very knowledgeable about this hormonal regulation, but as Simcha pointed out, it has to be related. So yes, it's an open venue of research, a very, very fruitful, probably, venue of research.
Thank you, Lieta, for your answer. I saw Kasia, you raised your hand at some point. Is that still true?
Yes, thank you very much. I have well, first of all, thank you so much to everybody who gave a talk. Really, really interesting. My first question or a comment is going back to the idea of quantitative wood anatomy. I came to this seminar asking, hoping to see that somebody would be talking about, oh, here and here there are this variation in proportions of, say, parenchyma or I don't know, dilution rates or fibers in, and talk about the function, so the link between the structure, anatomical structures and function. But I'm kind of left a little bit thinking that maybe we actually don't know much about this quantitative links. Maybe perhaps except for the talk by Jožica about the sizes of sieve tubed elements. And oh, start my video. Yeah, so I guess my question was, does anyone have information about the functional interpretation of quantitative bark anatomical features? That's one question. The other question is, does anyone know about how growth of bark might be related to bark anatomical features?
I will turn this question to Jožica first, because Jožica, in your
yes, it was, yes, I have similar question to Joyce, yes.
Yeah, because Jožica, you also mentioned we have many tools to do quantitative wood anatomy in the wood, which now it's probably time to apply them into the phloem. But why are we not doing that, or so little?
Yeah, well, Alan, you remember that during cost section, we had one working group trying to explore a bit more. Also, well, in our case, we are mainly now, we are focusing only on the youngest phloem part. And well, it's very time consuming, I have to say, so it goes very slow. And what we are doing now, we try to, because we have this multi-year data, 8 years, we try also to explore a bit more this climate growth relationship. But of course, we have idea or wishes that we go also to try to analyze the entire, maybe the first non-collapse and then collapse part of the phloem. Yeah, but it's sometimes I have also the problem, Alan, you remember that when first of all, the quality of the sections, if you're working on cross sections, need to be very good. Sometimes it's very difficult to distinguish in hardwoods between parenchyma cells and late phloem sieve tubes. And so there are some methodological maybe obstacles, but yes, it's I think that maybe it's time really to start to pushing also in this direction, because these image analysis systems, they maybe also enable us to analyze the crushed phloem parts as well, try to reconstruct, let's say, the size. Yeah, but yeah, it goes, at least in our case, it goes quite slowly. It's not, it takes time, it's time consuming, and if the bark or the phloem part, it's not good, then we need to repeat it and so on. And yeah, yeah.
Thank you, Jožica. And Marcelo, again, here we have also Jožica saying it's difficult to cut sections from the bark. Everybody is saying so. Is it really true, or?
Yeah, definitely. It's not easy. Yeah, there's no question. And every material is different, so you have to be trying and moving the shit, like the knife around until you get a good section. But yeah, hysteresin, usually it's easier to section, but the other methods are more complicated.
And there is, if I may just comment, we tried with ultrastructural observations. So we also, in one study with Peter, we studied the process of callose. in sieve tubes, but then it's another question if this, I don't know, if the sampling can also affect and so on. So they're living cells, and you need to be quite sensitive how to handle on, maybe even during the sampling, during this short time, you can cause, you know, some artifacts on the results. So I think it's, yeah, it's much more complicated.
I want to ask, in addition, Jožica, you showed some results, also Julieta, about bark and water stress. But under water stress, you would get also more collapsed cells. So how do you quantify it? How do you make quantitative anatomy of this?
Yeah, it was in the chat also one question, if this can be kind of irreversible process. I just got a visit 1 week ago in our lab where the colleagues from Czech Republic, they showed me images when they said that it can be reversible. I was not really sure, but then we are decided we'll try to explore a bit more. But from the seasonal data we have or cross sections, I never find such a trend in our sections, but it's one hand, it's also true that maybe it's not, I don't know, there were drought events, but maybe not so severe. But to see this pattern, I've never saw it, like in Norway spruce or in beech, our samples. But yeah, the colleagues, they showed me some images of Pinus sylvestris. So we decided that we will collaborate a bit more to see if this is true. But still, I think what we need to say also here, if we are talking about anatomical structure, we don't know how it is, if they're really conductive. So it's that's why I'm saying they potentially can be conductive. So we never try, you know, to go into this physiological part. But this is also something that we would like to do in the future.
Thank you. Joyce, your question.
Hi, so thank you so much to everybody, the organizers and the speakers. I'm kind of new to the community. I'm an assistant professor at Cornell. I study vine anatomy. And so it's good to finally meet some of the people that I read their papers or cite. But anyway, so I have 2 questions. They're both for Julieta. I wrote them in the chat, but maybe it's more of a discussion. So, a lot of the plants that we study have, well, they're all vines, and they often have successive cambia. So they'll have alternating rings of phloem and xylem, And something that comes to mind sometimes for me is like, why not have alternating rings of just within the xylem, just, you know, lignified xylem and then non-lignified parenchyma and then lignified xylem? Like, why intersperse the sieve elements and the phloem within the xylem? What kind of sort of functional hypothesis do you have about that? I'm just thinking about all the different ways in which the phloem functions outside of fire. And then the second question is about, it's kind of unrelated to the first one, but it's also about function of phloem. So another like line of research in my lab is thinking about tension wood. And so tension wood being the sort of wood that trees make to correct their posture, and they often produce gelatinous fibers to create this tension that can erect a prostrate structure into a vertical position. And I'm just curious about why that function seems to be relegated specifically to the xylem instead of to the phloem. I know there are some reports of phloem G fibers, but it seems to be really scarce compared to the xylem. So why would that function of like posture control be relegated to the xylem? And you can take either one or both, or in whatever order you feel. And nice to meet you. Thank you.
Thank you very much, Joyce, for your questions. It's very nice to meet you. I hope that in your next visit to Mexico, we can meet. And regarding the first question that has to do with successive cambia, we have measured bark in species with successive cambia, and as you say, it's very interesting that the disposition and what are the functional implications of that structure, right? And given that successive cambia are common in seasonal sites, in species of seasonal sites that are subjected to periodic drought, maybe there's an explanation there, because if bark is involved in the refilling of embolisms, as some studies have suggested, so maybe in in plants that experience embolisms as part of their yearly life. Maybe there's a selective advantage there. So, but I don't have an answer, a definite answer, and it's another open question. And regarding your question of tension wood and bark, there's a very recent study of the role of bark in the sort of things you're mentioning. I'm not very familiar with the literature, if there's any other paper, but I can send you that paper. And it seems that bark is also involved in this postural changes.
Are you thinking about Bruno Clare's work?
I think so. I don't remember the authors, but it might be
okay. I really
send it to you.
The answer to the first question about like embolism repair, that kind of makes sense because of one of the plots that you showed that the phloem is going to have more soluble sugars than the xylem in the twigs and the bark, and excuse me, the twigs and the trunks and in the roots
and the roots
and in the roots, and so maybe it's doing a better job of is doing a better job than would be the xylem parenchyma as far as embolism repair in terms of having the soluble sugars availability. So maybe it's just a better, okay.
Maybe just I comment this tension wood because I also saw in the chat. Well, we sometimes saw a lot of tension wood, especially in branches, in wood. So we immediately knew that we need to exclude this kind of samples from our observation because we need to follow normal dynamic. But also in this case, what we saw is that the structure and the phloem was different. So it was also the increment was wider, and also the structure was anatomically different. Usually it was more of the late phloem, and the size of the sieve tubes also seemed to be different, smaller. We never actually measured because we always exclude this kind of samples. We are not interested in this part, but we from the structure of the phloem, we saw that there is tension wood. I didn't need to look from the tension wood in the wood. So yeah, I think there are anatomical adjustments also in the phloem.
Yeah, right. There's very few papers on that, but yeah. Thank you.
Great discussion here. Reza, you also have a question, right?
Yeah. Thank you for holding this seminar. It was really interesting. I have a question. Maybe Marcelo can answer, but anybody else who has experience maybe can help answering this question. Regarding wood quantitative anatomy, along with the cross-section analyzing, for example, the size of vessels, we sometimes use macerated wood. We do maceration and do some quantitative analysis on macerated fiber, different fibers, or the vessels, and etc. I was wondering whether there is similar experience regarding phloem or bark. I mean, did somebody macerated bark, or maybe it may complicate the stuff more than rather than making it easier. I mean, can it help to do get some quantitative data out of the bark, or maybe it's already too complicated to have macerated cells a lot? Thank you.
May I answer that? So yeah, we do macerations for phloem and pediderm, and it works just fine. But yeah, we didn't suggest that when we were discussing the IAWA committee. I think because many of the cells are non-lignified, we were concerned how much they would change in the maceration in their morphology, how much they would be crashed and crushed. So, and that's why we decided to make the measurements using the longitudinal section mainly. But of course, you could, I think it would be a good idea to use the macerations for the fiber length if you wish to measure fiber length, because then you're talking about a lignified cell and then it's easier to do it. But yeah, you can do, but you have to be, it's not as easy as wood because you have to be more you have to understand that it will be quicker than for wood, so you can't forget it there, you know, and then and expected all the cells to be there, so you have to use shorter times.
Thank you. We have a question for Alexei, which comes from the chat. And the question is, Alexei, can we use bark macroscopic structures to identify plants as we do for wood? We do wood macroscopic identification. Can we do bark macroscopic identification?
Theoretically, yes, but you know, the problem is that there is no commonly accepted terminology for description, and there are several attempts to make such terminology for some plant families, and the bark characters sometimes are included in the identification keys. But the modern now I don't think that we can identify the use the microscopic characters for identification. About microscopic, bark is very poorly explored in comparison with wood. That's why I have done. Of course, if we know something about the plant, about the family, sometimes it helps. But it is wood is much better known than bark. That's why it is in the future.
Mm-hmm. Okay, thank you. Mariam, I see you have your raised hand. Is it a quick question?
Yeah, good evening, everyone. Thank you so much, the organizers of this program. It's quite interesting and educative. Hello?
Hello.
Can you hear me?
Yes.
Yes. Please, just a quick question. I've actually worked on bark and wood anatomy of South Africa Meliaceae, and the results seems to be quite informative, also with taxonomic importance. But I would like to ask us if there's anyone that can shed light on how to relate the bark, probably maybe the bark anatomical features, to biological activities of these plants.
What do you mean with biological activities?
Like, is there any relationship with the bark anatomical activity of plants to their biological activities?
What is the biological activity of a plant? Anything
biological
specific?
Activity of a plant. Probably maybe you are trying to look at the antimicrobial activity of the plant, or their toxicity activity, or the
medicinal properties, like medicinal plants?
Yes, please, medicinal plants. Does this bark anatomical descriptions or the wood anatomical descriptions, does it have any effect on these medicinal properties of these plants? Thank you.
Anybody to answer this question? Marcelo?
I don't know if the anatomy relates to some medicinal use, but of course, many barks have a very important role in medicinal use, and also in I think when I was at the Smithsonian, I remember that also not only medicinal use, but also in the construction of some Native Americans used basswood for making some of their tools and some of the things that their clothing. So yeah, but and in this case, in the case of the construction of clothing, then the anatomy was important because the basswood has these bands of fibers, and that was the part that was used. Here in Mexico, I know that also papel amate, which is a type of paper, is done by ficus. You macerate the ficus and make the paper. And probably in medicinal uses, probably the substance of interest will be in a cell, of course. Then the anatomy can be important to understand where the substance is coming from. So that's my idea, but yeah.
I would also add to that there are particular cell types that you can observe and at least correlate with something you might know about the secretion from that plant. So there are like laticifers that you can identify that look like something in the wood or in the bark, and then you can actually snap the stem and see the latex coming out. So you can identify that kind of stuff. Or oil cells, these are cells that actually have a anatomical signature that you can also look at the plant and see the oil coming out. So there are some types of cells that are not, that you don't have to, that you can just observe actually. So I would say luticifers and oil cells and there are many more. And I would refer you to the IAWA documents for that actually. If you go to the IAWA website and look at the bark list and the wood lists, the hardwood and the softwood list, you'll find more features there.
I'm terribly sorry, we have to interrupt this lovely discussion here because of time constraints. So Guillermo and everybody else, please feel free to use the forum to post your questions and answers. And I'm now handing over.
We can add the discussion in the forum too, and then people can continue the discussions there actually.
Yes. Now I hand it over to Georg for the closing remarks.
Okay, thank you, Alan. I just need to share the other screen. Here we go. Can you see that?
Yes.
And I turn on the presentation mode. Okay, now it's the other way around, right? Okay, here we go. Now you should see it properly. So a few words from my side. First, as we have heard, there is many unanswered questions, which is clear. So we heard from Alan and Ilana that yes, please use the forum. So you know, maybe there is questions popping up later on, or some unanswered questions in the chat. Although I have to say the chat today was super hot. So really thank you very much for your very active participation, and also to the presenters for their trying to reply as much as possible. That's really great. But maybe there is still more to do. Now, of course, this is not the last event that we will have. After the workshop is before the next workshop, which will be already the 5th. It will be in about 6 months. We don't know about which topic. So this is exactly also where we make a call to all of you. Basically, the reason why we have the topic of today's workshop is thanks to Ilana. And you also see where that led to. So she's now one of the coordination team members, right? So that's also, of course, an opportunity for all of you. Then how can you contribute from your side besides just making, as I said, or sharing your ideas for a next topical workshop? Well, I use the forums, but then, as I mentioned in the very beginning, we have this seminar series. And just don't forget about this. So if you think you have an exciting study of maybe general interest, or maybe also more specialized, narrow, whatever, but worth it to share with others, and where you maybe want to have some input or interactions, please approach us. And well, then we also have in the member area a online resources database, which is growing, which we are also about to restructure. And also there, of course, we are very happy if you have like a movie, right? We have heard about movies from Yoshitsa. Maybe that's something we could link there as well. So it's not a content just made for QNet. We are very happy to just offer the platform to make it more noticeable to the community, right? So please approach us and make suggestions. Or if you have anything else, well, then the QNet contact form, of course, or an email to a QNet coordination team member can be used. Now, in terms of, well, you see sometimes some of us have this nice background. So this is the small reward. Everyone who ever contributed will get for free, I say, this nice background, and you are entitled to use it for any workshop or seminar. And I hope actually by the end or in the future, more and more of you can use this background. So that's the new the incentive we can offer, right? Okay, so then with this, it's time for the big thank you. So first of all, to the presenters who really, I think, stimulated our thoughts now, but probably also in the future. So thank you very, very much. I would also like to thank very much to the organizers, so Alan, Ilana, and in the background, Angela, and then of course all of you for participating. Just to also remember again or remind you, so there is the recording. It will be available online in the QNet member area. We will send out an email to announce when this will be available, and potentially also the slides from the presenters or presentations. If the presenters agree, we will also upload there. So that's all from my side. And with this, I'm already looking very much forward to the next QNet activity. Thank you very much, and have a nice evening, morning, or night, or whatever it is in your time zone. I should also mention, well, we had at the peak 126 participants. And if I count some more, because some shared screen, I think that's really a very, very successful workshop. So thanks a lot and stay healthy. Bye-bye.
Bye-bye.
Bye.
Yeah, bye everyone.
Thank you very much.
Bye.